Hello, everyone. Thank you so much for joining us today for the first ever May the 4th Channel Be With You webinar event from Menarini Silicon Biosystems. My name is Bamboo Dong, and I'm a Scientific Affairs Liaison at MSB. We're very happy to have you all here today, and we're very thankful for the speakers who have volunteered their time and expertise to share their work with us. Today's event will be dedicated to the CellSearch open channel, which takes advantage of user-defined biomarkers to harness the full power of the CellSearch system for rare cell enumeration. Over the next 2 hours or so, you'll be hearing from an incredible lineup of oncology researchers and clinicians who have been doing amazing and groundbreaking work using the 4th channel on a variety of disease states. You'll learn about the biomarkers they used, the different ways that markers have been used to stratify disease progression and monitor patients, and what this can mean for the future of precision medicine and patient care. We'll also be hearing from some of MSB's own on topics like our global lab services, how the power versatility of CellSearch can be just one blood draw away, and a quick look at the newest member of our product family, the CellMag. If you have any questions for any of the speakers, please enter them in the chat box on your screen. For the sake of time today, we won't be able to do live Q&As, but we will make sure to forward your question to the presenters and make the answers available after today. If you're having any technical difficulties, please let us know in the chat window as well, and we will do our best to sort it out for you. For the best audio experience, we do recommend using your computer or device's built-in audio instead of the phone dial-in option, as there are some features that may only work through your device's speakers. I will do my best to try to move the event along as quickly as possible, as I know we have attendees from different time zones all around the world. And with that, I will stop talking and introduce our first speaker, Dr. Filippo Dall'Olio from the University of Bologna. Thank you, and may the 4th be with you.
Thank you very much. Okay, perfect. My name is Filippo Dall'Olio. I work in the University of Bologna in Italy. I'm a PhD student, and I wish to thank the organizer for having invited me to present my study. My study is about PD-L1 in circulating tumor cells as a prognostic biomarker in advanced non-small cell lung cancer treated with immune checkpoint inhibitors. Briefly, CTCs are a promising source of biological information in cancers. They are also related to tumor burden, and there is an increasing number of evidences about its role on immune response against cancer. For now, PD-L1 is the only validated biomarker for immune checkpoint inhibitors in lung cancer, and data on the role of PD-L1 expression in CTC are still lacking. So this is a prospective single-center cohort study. We enrolled advanced non-small cell lung cancer patients. We received second or third line anti-PD-1 or PD-L1 monotherapy. Blood samples were obtained within 14 days before treatment start. CTCs were identified and counted with CellSearch system, and PD-L1 assessment on CTCs was performed with the PE conjugated anti-human PD-L1 antibody clone MIH3. We divided patients in 3 categories: no CTC, so CTC null, at least one CTC, and at least one CTC PD-L1 positive, and they are CTC pos, and at least one CTC and no CTC PD-L1 positive, CTC neg. The primary objective of our study was to evaluate the correlation between CTC abundance and PD-L1 expression on CTC with overall survival, while the secondary objective were to explore the correlation between PD-L1 expression on CTC and PD-L1 expression on matched tumor tissue, the correlation of CTC count and PD-L1 expression on CTC with PFS, progression-free survival, response rate, and durable clinical benefit, and the correlation of CTC number with tumor size. We enrolled 39 patients. CTC were detected in 24 of them, ranging from 1 to 59 cells per blood sample. Of them, 15 has no cells, 13 has CTC positive, PD-L1 positive, and 11 CTC PD-L1 negative. The three categories were very similar for all the clinical pathological characteristics, apart for gender, that with the CTC positive were more common in female patients, but the low sample size did not allow us to draw any conclusion on that. We find that there is no correlation. We found no correlation between PD-L1 positivity on tissue and on CTCs, and even no difference in terms of PD-L1 productivity TPS on tissue between those with CTC PD-L1 positive and those with CTC PD-L1 negative. Anyway, none of the patients with no PD-L1 expression of CTC has a PD-L1 more than 50%. We found instead a correlation between CTC number and baseline tumor size, as expressed by the sum of diameter according to Ricci's criteria. And in terms of overall survival, after a follow-up of 12 months median, we found that the CTC negative, a patient with no PD-L1 expression on CTC, has 2.2 months of median overall survival versus 3.7 months for those with PD-L1 positive CTC and 16 months for patients with no CTC detectable. We can see the curves, and we can see that there is the possibility of long-term survivorship. That is one of the reasons for the use of immunotherapy in lung cancer. There is none of the patients with PD-L1 negative circulating tumor cells had an overall survival more than 5 months. And instead, we found a little, if no difference between those with no CTC and those with PD-L1 positive circulating tumor cells. We validated it with a multivariate analysis that included all other well-recognized prognostic factors, and found out that our CTC score is still significant when accounted for other prognostic factors. Similar results we obtained with progression-free survival. No difference between those without CTCs and those with PD-L1 positive CTCs, and a bad outcome for those with PD-L1 negative circulating tumor cells. And the prognostic value was confirmed also analyzing response rate and durable clinical benefit. So in conclusion, our paper confirmed the prognostic validity of CTC's detection in blood sample and its relation with tumor size. It also suggests that PD-L1 assessment on CTC could add a significant prognostic value for non-small cell lung cancer patients receiving immune checkpoint inhibitor therapy. We found no correlation between PD-L1 expression on tissue and on CTCs. And as a future ideas of development, future analysis on larger set of data is warranted to investigate the predictive value of our score, meaning we should enroll patients treated with other agents to better understand the predictive value of this score, and potentially patients treated with combination treatment like chemo IO for lung cancer. That is an option now, even for patients with high PD-L1 expression. Anyway, assessment in first-line treatment is ongoing. Thank you very much for your attention.
Thank you so much for that wonderful talk, Filippo. As a reminder to attendees, if you have any questions, please input them into the chat window, and we'll make sure to get them to the speakers. I am going to introduce you to our next speaker, Dr. Jaco Kraan from Erasmus University Medical Center in Rotterdam. Let me go ahead and, whoops. There we go.
Okay.
Dr. Khan, you should have the controls.
Let's try if I can show my screen.
Perfect. We can see you.
You can see it now?
Yes, we can.
Okay.
All right. Thank you.
Okay. Yeah, thank you for the kind invitation for this webinar to present some of our experience. As you said, I'm Jacob Kraan, and I'm a postdoctoral researcher in Erasmus Medical Center Cancer Institute in Rotterdam, the Netherlands, working with the CellSearch already from 2005. And I'd like to share some of our experience with the 4th channel. I would like to shortly present one of our studies with the commercial HER2 marker. I would like to show how we use the 4th marker as an exclusion marker for CD146 enrichment, or MCAM, and some of our recent work using PSMA, FITC, conjugated antibody in a clinical prostate cancer study. First, one of our studies using the HER2 in the 4th marker. Here we try to investigate the prognostic impact of HER2 positive circulating tumor cells in patients with HER2 negative primary tumors receiving endocrine therapy. And additionally, we also study the prevalence and prognostic significance of these HER2 positive CTCs in a chemotherapy cohort. As said, we included metastatic breast cancer patients with one or more detectable CTCs starting a new line of treatment. The CTCs were enumerating using the CellSearch system and characterized with the HER2 antibody, the HER2 phenotyping reagent. The primary endpoint of this study was the progression-free rate after 6 months of endocrine treatment in HER2 positive versus HER2 negative CTC patients. We used the scoring system very well addressed by the group of Riedorf, published in the Clinical Cancer Research in 2010, where you have a 3+ score with this bright HER2 staining with no background, up to no scoring, a 0 score with no HER2 staining at all. These are the characteristics of our studies we found. We found over the overall 29% patients with HER2 positive CTCs with a staining of 2+ or 3+ or 2+ or more. And the progression-free rate after 6 months did not differ between the endocrine therapy arm and the chemotherapy arm. This is also shown by the Lever-Jennings plots for both the endocrine therapy and the chemotherapy for progression-free survival and overall survival. As you can see, however, we observed a trend in a worse prognosis in patients with HER2 positive CTCs present. We did, however, unfortunately didn't find a significant difference, and therefore we had to conclude that there was frequently occurring HER2 stats between CTCs and the primary tumor, but we couldn't find a prognostic impact in our metastatic breast cancer patients cohort, however. The next thing where we're using the 4th channel for was as an exclusion mark in a study where we used MCAM or CD146 magnetic enrichment on the CellSearch. And that was initiated by one of our studies where we studied the epithelial adhesion molecule expression on our cohort of breast cancer tumor cell lines. And there we observed that in the normal-like intrinsic subtype cell lines, the EpCAM expression was lower, absent. This also here shown in the representative flow cytometry graphs, where you have both the EpCAM expression on basal-like and luminal cell lines, but not on a normal-like cell lines. Therefore, we investigated if we could add additional markers for CTC enrichment using the CellSearch. And we were looking for candidate markers based on gene expression data and also showing a high membrane expression on EpCAM negative cells. And of course, they should be absent on hematological cells or easy distinct by additional markers. We came up with 3 potential markers: caveolin, mucin 1, or the melanoma adhesion molecule EMCAM or CD146. These were already available from for the CellSearch and bound to ferrofluids. And as you can see, this expression of CD146 is very well expressed on all of almost all of our cell lines, breast cancer cell lines. The CD146 marker is also expressed in endothelial cells, and therefore we use in the 4th channel CD34 as an exclusion marker. So not together with CD45 as been done in the melanoma kit, because the cells are CD146 is only also expressed by melanoma cells, smooth muscle cells, but that will be a problem. Dendritic cells and activated T cells, a subset of activated T cells, but we can exclude these with the CD45 exclusion marker. And to exclude endothelial cells, we use CD34 that's highly expressed, as shown here by flow cytometric data on the endothelial cells here, sorted and positive for Willebrand factor and having the typical endothelial morphology. So in this way, we could exclude circulating endothelial cells using the CD34 in the 4th channel and make a good distinction between circulating tumor cells and circulating endothelial cells. And we also had the advantage that we could also enumerate the circulating endothelial cells by analyzing on DAPI plus CD34 plus cells. And then you can see that a subset of this endothelial cell also express cytokeratin on a certain level, especially with cytokeratin 8 and 18 is known to be expressed by endothelial cells. This approach was then used in a clinical trial where we combined EpCAM and EMCAM CellSearch enrichment to increase the CTC capture. That was our goal, and therefore we did 2 countings, one on EpCAM and one on EMCAM. We did it before the start of the first treatment and after the first cycle of treatment. These were the results in our study. We had a CTC count of 1 or more CTC in 90% using only EpCAM, 12% in using only EMCAM, and combined we had a 25% CTC count greater than 1 or greater, and so we increased the CTC capture from 90 to 25%. And remarkably, 5 patients had no EpCAM positive CTCs, but they did have EpCAM positive CTCs. And after the first cycle of treatment, this was actually increased. We had 90% only EpCAM, 60% only EMCAM CTC count, and combined we added up to 30% of CTC count. So here we increased the CTC capture from 90 to 30%. And again, we observed patients with no EpCAM positive CTC, but with EMCAM positive CTCs. So we concluded in the study that the majority of normal-like cell lines, so not CTC, but cell lines do not express EpCAM. They do express CD146 or EMCAM, and we slightly increased the CTC detection in stage 2-3 breast cancer patients, combining these approaches. However, it was, of course, we had a low number of CTC positive patients in this cohort with a relatively low, small number of CTCs. So maybe this has a higher impact in the metastatic setting. The last part of my talk, I want to show some recent work where we examined the PSMA, the prostate-specific membrane antigen expression on circulating tumor cells using CellSearch in prostate cancer patients. PSMA is expressed by normal and malignant prostate epithelial cells, also on the adenocarcinoma cells, and on vasculatures of other malignancies. We first examined the PSMA expression on our cell line cohort, and here you see the very high expression of PSMA on the LN cap cells, and down titrated, and you don't see any background fluorescence in the normal range of 2 to 0.5 microgram per milliliter on white blood cells, and we either didn't see any expression on other non-prostate-like cell lines. Then we did different titration experiments on the CellSearch using different concentration of the PSMA, using different times of exposure. And here you can see the expression of the PSMA on spiked LN cap cells. And importantly, of course, we also had our negative controls where we did no stain control, and we didn't see already some small fluorescent background, but the cells are negative here. And importantly, we didn't see any expression on the leukocytes of the PSMA. And now this is a running study where we do use this PSMA marker on patient samples. This is an example of a patient sample where we see CTCs, actually almost all expressing PSMA, only you see some cells with a very dim level of PSMA. Well, actually all EpCAM positive, CD3 to 6 positive cells express PSMA in different levels on the flow cytometry plots. So it looks like the CellSearch is not able to detect the low expressing cells very clear. So to finalize, I think the 4 channels can reliably detect antigen markers that are highly expressed by circulating tumor cells. We also have experience with M30, K167, and PD-L1, but I expected it to be also already addressed by the other speakers. And I think for the future, increasing the sensitivity of this channel, using an other light source, other filter combinations, or a better camera detector, can further improve the detection level of antigen markers on CTCs in the 4 channel. Of course, I'd like to thank my colleagues and co-workers making this all available, and I'd like to thank you for your attentions, and I'm happy to take any questions, and I understood it will be by the chat. Thank you.
Awesome. Thank you so much. That was excellent. If anyone has any questions for Dr. Khan, please let us know in the chat window. And we're going to learn about Menarini Silicon Biosystems' newest product, the CellMag. Here to walk us through the technology is Scientific Affairs Liaison, Dr. Cameron Lilly.
Hello, my name is Cameron Lilly, and I'm a Scientific Affairs Liaison with Menarini Silicon Biosystems. I'd like to take a few minutes to talk to you today about something that we're very excited about, and that is a new line of products available from us for the isolation of CTCs from blood, the CellMag product line. The CellMag is really designed to streamline the manual immunomagnetic enrichment and staining of cells using the established gold standard ferrofluid technology that supports the CellSearch, from whole blood all the way through to enriched EpCAM positive CTCs. This is a very simple, easy to use process. Sample processing with the CellMag is very similar to that for the CellSearch. 7.5 mils of blood are brought in from a cell safe tube, buffer is added, sample is centrifuged, and the plasma is aspirated. At this point, ferrofluid is added, and there's magnetic incubation to isolate the cells that you want and stick them to our magnet. Fluid in the tube will be aspirated, removing any of our unwanted cells. At this point, the remaining cells are washed, and then permeabilization and staining reagents are added to the tube. The result is stained, fixed, labeled target cells with some contaminating leukocytes that are counterstained for as well. So what is it that makes our ferrofluid so special? There are lots of immunomagnetic enrichment technologies. Why is ours the gold standard? Well, at its core, the bead is an iron nanoparticle with a polymer layer, but what makes it special is what's on the surface. First, of course, there are the anti-EpCAM antibodies, which are responsible for recognizing and binding to EpCAM on the surface of our CDCs. But included in this is also a biotin analog, and this is responsible for amplifying the amount of ferrofluid which will attach to one of our cells, which drastically increases our enrichment. We'll see that in a second. So if we have a CTC in solution and we add our anti-EpCAM ferrofluid to the CTC, the first thing that happens is that this ferrofluid will bind to the surface of our cell with the anti-EpCAM antibodies recognizing EpCAM on the surface of the cell. It's at this point in the process that streptavidin is added in solution. The biotin analog on the surface of the beads will bind up this streptavidin, and they will then bind the excess anti-EpCAM magnetic ferrofluid nanoparticles through this network of biotin analog and streptavidin binding, and this increases the volume of iron nanoparticles around our target cell. So now we have a good strategy for enrichment. How then do we get the cells back? Well, this is very simple, and it takes advantage of competitive binding kinetics of biotin, our biotin analog, and streptavidin. So to this mixture and this mesh network of magnetic ferrofluid particles, biotin is added, and it will competitively bind our streptavidin. When this happens, the network of nanoparticles that has been built around our cells is effectively washed apart, and what remains at the very, very end is our CTC isolated, coated with our anti-EpCAM beads. And then, of course, just as is the case in our CTC kit, cells from the CellMag kit are immunofluorescently labeled. CTCs are labeled with an anti-cytokeratin PE antibody, and of course, DAPI to stain the nucleus. And leukocytes can be distinguished by being, of course, DAPI positive, but also binding to anti-CD45 APC antibody, which is included in the kit as well. So what products are available? Well, right now, we've launched a suite of products for the isolation of epithelial CTCs. We have the CellMag magnetic tool, of course, the CellMag epithelial CTC kit, as well as the consumables necessary to run this isolation. Down the road, we do have other kits that we are in the process of optimizing and launching. This will be a pan-CTC kit, so CTCs from more lineages, a CXC kit for further characterization of your cells of interest, a profiling kit, and a circulating endothelial cell kit. So about how long does this take? Well, this optimized protocol is not particularly long. Sample preparation takes 30 minutes, and then the magnetic separation of your cells takes 40 minutes. Washing steps there take 15 minutes, and then permeabilization and staining takes 50 minutes. Following that, a 20-minute fixation is required, and the total time to process a sample, which includes staining and preparation, is 2.5 hours. So the advantages of this is that the enrichment and staining are done in a single step. There is no need for additional staining steps after the isolation of your cells. It's a standardized aspiration. This is very important when sample to sample and investigator to investigator variability needs to be reduced. Because this is designed to be run from whole blood, your samples are stable for up to 96 hours in our cell-safe tubes. And of course, this brings the gold standard ferrofluid aggregation technology that backs the CellSearch into your hands at a fraction of the cost. CellMag is extremely simple and easy to use, and there's no additional training required to operate it. For more information, please check us out at siliconbiosystems.com/cellmag. Thank you.
All right, thank you so much, Dr. Lilly. And next up, we'll be hearing from our own Sarah Lazaro, Head of Clinical Lab Services and Operations at Menarini-Silicon Biosystems.
Hello, everyone. As Bamboo just mentioned, my name is Sarah Lazaro. I have the honor to work with the scientists in MSB as the Global Head of Clinical Lab Operations and Services. I want to thank the organization for giving me the chance to talk about what we do in the lab services as part of the Menarini Group and what we can offer in terms of the open channel for anybody who may be interested in our services. So I want to start the presentation giving a brief overview of MSB, our different platforms that we use to provide services, what can we do in the lab for you, and finish up with some examples of real patients and what we've been able to do for the open channel working with customers on specific biomarkers. So as you can see on the slide, this is a very brief overview of different milestones for our company. In 2004, the CellSearch obtained FDA clearance for IVD use. We were acquired MSB by Menarini in 2013, and also a very important milestone for us was in 2014 when the Parade version 2 was launched, and we are ready in 2021 to offer the Parade Plus. So the lab team, we try to what we want is to support our mission, the mission of MSB, of rare cell analysis with single cell precision by offering global laboratory services. So we accomplish that by processing samples into different locations. One is in Pennsylvania in the US, the other one is in Italy, and that allows us to test and receive samples from all around the world. This slide shows a very brief and summarized picture of our workflow. Everything starts with collecting a sample by a very non-invasive method that is a simple blood draw. For that, we use our cell-safe cell rescue tubes, and I'll talk a little bit more about them in the next slide. This is a very central and important part of our workflow, as I will describe a little bit later. So once the sample is collected, it's been shipped to the lab, we receive it there, and we do the testing and analysis. We can provide results in the form of the report that you see on the slide in a couple of days. This is being done with our CellSearch with the CellSearch platform, but we can also offer downstream applications. So for that, the sample can be freezed, and we can work on different options and also offer results in fairly quick turnaround time. As I mentioned before, very important part of what we offer in our workflow in the lab is the self-safe tubes. We are also offering, or very close to offer, the self-rescue tubes that are more focus on RNA applications. So what the self-safe does for the samples is that through a proprietary preservative, cells can be stored for 96 hours at room temperature. As you can imagine, that is very important when you work with global services. So that allows for shipment of samples from remote locations all around the world, which is not very common in some other lab services. And it also improves the reproducibility and reliability of the CTC analysis. We are actually working on even increasing the 96 hours, but that's a working process. I want to take just a few minutes to describe the two platforms that we use in the lab service team. The first one is the CellSearch that offers the additional channel we are talking about in this seminar today. So this is the platform that is being used for the enumeration of circulating cells. The blood collected in the self-safe tubes, self-rescue tubes is being processed with the CellSearch reagents in the CellTrax autoprep system. That is an automatic system that that allows the preparation of the sample and make it ready for analysis. The sample is enriched and transferred in the cell magnet cartridge holder, and then is being analyzed in the CellTrax analyzer, where the images are presented to the operator for classification. As I mentioned before, we can offer downstream applications with the depth array system that allows for cell sorting to the individual cell with 100% purity. And this slide shows the three most important components of the system, which are the instrument itself, the depth array. Everything is being performed in the depth array cartridge, and then the software, the cell browser software, allows for imaging, analysis, and interpretation. I'm gonna move towards talking a little bit about the lab services, what we can offer, what we do for our customers. We have enough operational capacity in both of our labs to work with all kinds of customers. We have a fairly amount of instruments validating for CellSearch and depth array projects, but we are also working on setting up and offering more molecular options for customers that may be interested on that. We are accredited by different agencies, and our main focus right now is to work with biopharma customers in clinical trials, anywhere from phase 1 to phase 3. In the US, we are CLIA certified and ISO accredited. We also have licenses for different states. And in Bologna, we are mirror labs. We follow the same quality management system, working on getting ISO certification, hopefully in the second half of 2021. And in this case, we work also very closely with different customers in Europe to offer what we call fee for service, which is a way to prove concepts and demonstrate the capabilities of our services and our platforms. Everybody working in the lab services team has extensive experience. We can offer more than 50 years of clinical trials expertise, serving major pharmaceutical and biotech organizations. We have a proven record of success with quality audits, not just with regulatory agencies, but also audits that are being performed by our customers. Have to say that those are more intense and more in-depth probably than even the accreditation agencies. Everybody in the lab is highly experienced. We have all kinds of expertise, from engineering to lab sciences, medical doctors, PhDs, and something that is very important and we feel very proud of is that the scientists who develop the CellSearch and the array systems are still part of the team, and they work very closely with us, R&D and the QC team. To kind of put everything in perspective about our extensive experience and offer some information, in the more than 15 years of experience offering CellSearch applications, we handle more than or close to 25,000 samples. We've been part in about 60 clinical trials, partnership with many pharma companies, and we've gone through more than 40 successful external audits. And this is more related to what the topic of today's seminar is, and how we can offer or tailor the customer unique needs. We are, as I mentioned before, highly qualified. We are certified by agencies, but we also perform our testing using GCP and GCLP regulations. We can tailor our services to what the customer needs. We work very closely with our customers to make sure that the studies are expedited. Any issue that is happening with samples during shipment or processing is immediately communicated to the customer for rapid resolution. We understand how patient samples are for all of our customers, and that's very important and crucial for us. And we are scalable in our current capacity. We can work closely with different kinds of sample sizes for studies. So one of the things that we offer as part of our services is biomarkers using the 4th channel. We have some options that are already available in our menu, but we can also work with customers to build or to develop what they need. So the way to make this work is to either use one of the options that we have available in the MSB sales service menu, and or also work with us, reach out to the lab services team. We work very closely with our R&D department to tailor our offering to the customer needs, and we partner with you. So the next few slides, and the way I want to finish the presentation is showing images of real patient samples that are part of studies that we are working on currently, and how the 4th channel is being used. So this first one shows PDL1 images. So on for this slide and the 3 or 4 slides that are coming, the left side of the slide is going to show the positive marker. The right hand side of the slide is going to show the negative marker. So as you can see, for PDL1, we can clearly differentiate samples that are positive, as they are shown in this area of the slide, and it's also very clear when a sample is negative for the marker. Okay. So the same can be shown. That was my idea, to show you some other markers, ER and HER2, I believe. I still cannot make this work. So the images that were part of my presentation are very similar to what you are seeing here. And something that I also would like to highlight is that our future for lab services is to move towards a centralized model, where instead of having a focus on selling instruments and reagents, we want the customer to send the samples to us for different applications. One of them hopefully will be offering custom biomarkers. And with that, I just want to thank you for the opportunity to present our work with the 4th channel and what we can do for you in the lab service team. Thank you very much.
Thank you so much, Sarah. I'm glad we could mostly get the technology sorted out, and we greatly appreciate your time today. Some of our next speakers couldn't be here with us live today, so they were gracious enough to record their presentations ahead of time. As a reminder, we do recommend using your computer or device's speakers for the best audio experience. And with that, I am pleased to introduce Dr. Elisabetta Rossi from the University of Padua and the Veneto Institute of Oncology, IRCCS.
Good morning, everyone. Thanks for having me. What I want to do today is to put the spotlight on CTC subpopulation using the 4th channel of the CellSearch platform. Tumor heterogeneity describes the difference between tumors of the same type in different patients and between cancer cells within a tumor. Both can lead to different response to therapy. Liquid biopsy is a powerful tool to monitor and predict tumor recurrence and therapeutic response of patients. Circulating tumor cells have been shown to be good predictors of metastatic progression and therapeutic efficacy. Circulating tumor cells can be genetically and phenotypically quite heterogeneous. Each subpopulation may contribute differently to metastasis, therapy resistance, and the consequent clinical course. However, the differential contribution of this subpopulation is not fully understood, but we think that it is important to understand this difference in precision medicine era. In fact, every subpopulation can allow us to identify high-risk population or to prevent disease relapses, or to identify patients with a higher benefit from a specific treatment. All these give more time and better quality of life to the patients and save money for the national healthcare systems. Over the past 15 years, we have developed several additional markers to phenotypically characterize circulating tumor cells and melanoma cells. In the last year, we try also to characterize circulating endothelial cells. On the left, you can see a list of additional markers that we currently use in our lab. The hallmarks of cancer are present in all cancer cells. These also include apoptosis evasion. The loss of apoptotic control allows cancer cells to survive longer and give more time for the accumulation of mutation. Mutation can increase the invasive ability, deregulate cell proliferation, and interfere with differentiation of cells and induce angiogenesis. Characterizing the viability of CDCs in each patient may be important to improve prognostication and individualized treatment. In fact, the presence of exclusively apoptotic CDC in a patient may represent a favorable prognostic factor, whereas the preponderance of proliferating cells could be related to poor patient's outcome. We assessed the apoptotic status on CTC in patients with early and metastatic cancer. In order to reach this purpose, we use two different additional markers. The first, M30, only for epithelial cancers, and the second, gamma-2AX, only for non-epithelial cancers. We found that in the early phase of cancer, there are a high percentage of apoptotic CTCs. When we move on to the late stages, we have two different behaviors. In prostate, breast cancer, and non-small cell lung cancer, we found that the number of apoptotic CTCs is very low. On the contrary, in colon cancer, kidney cancer, and melanoma, we found a high level of apoptotic circulating tumor cells. We think that it is important to know the baseline of apoptotic level before to start a treatment. Another use of the fourth channel may be to highlight therapeutic targets in CTCs. In fact, we set a specific target in order to show the presence of EML4-LK protein fusion. On the left, you can see that our test well recognizes circulating tumor cell positive for EML4-ALK and circulating tumor cell negative for this target. On the right, you can see that this test could be used in order to monitor the specific treatment, but also allow us to stratify non-small cell lung cancer patients. In fact, this test highlights patients with a very short progression-free survival. Researchers are known to be skeptical and curious, and for this we checked whether the drug binds CTC and where. In fact, we set a specific test in order to know if trastuzumab, a monoclonal human antibody used to treat HER2-positive breast cancer, if this drug could recognize CTCs with high and low intensity of HER2. As you can see, this test identified that trastuzumab recognized both high and low intensity HER2 CTCs, and that recognized also the point on the membrane in which the receptor bound the drug and stopped the signal transmission. We also used the fourth channel to study the efficacy of drug against bone metastases. Recently, evidence suggests that rank ligand rank signaling influence not only microenvironment of bone, but also could promote bone metastases. Previously study demonstrated also that rank expression in primary breast cancer patients were associated with poor prognosis in terms of metastatic bone relapse. Denosumab, a monoclonal human antibody against rank ligand, represents to date the gold standard for preventing skeletal-related events onset in bone metastatic patients. Our data highlighted that the effectiveness of denosumab depends on the expression of rank in CTCs. On the left, you can see two typical patients treated with denosumab. In a patient's good responder. In fact, during all period of treatment, these patients had rank-positive CTCs. In B, a poor responder. In fact, during the treatment, only in the first 7 days, these patients had rank-positive CTCs. This result highlights how the oncologist could decide to stop the treatment when the patients don't respond. And the oncologist could decide after only 7 days to change the treatment in order to avoid the side effects of the drug. We also study CD44V6. Why? Because CD44V6 is a CD44 isoform, mostly involved in cancer cell migration and invasion. And as we demonstrated that CD44V6 could also be used as a stemness marker. In fact, CD44V6 contributes to chemoresistance in colon cancer. It's noted that these isoforms are involved in the reduction of apoptosis, in an increase in proliferation of cancer cells, in the induction of multi-drug resistance, and also in the induction of autophagy when the cancer cells have to overcome specific stress. Our study in a colon cancer cohort demonstrated that CD44V6 seemed to act as a drug-resistant signature in CTCs, and it might suggest stem-like properties. On the right, you can see a very good picture where we highlighted that CD44V6 is present in high expression and also in medium or low expression CTCs. During the past 10 years, targeting cancer metabolism has emerged as a promising therapy for the development of selective antineoplastic agents. It is important to stratify patients that could benefit with a chemotherapy targeting metabolism. To this purpose, we study 126 HER2-negative non-diabetics metastatic breast cancer patients at the first evidence of disease relapse, and we decided to use as a prognostic and predictive marker the expression of insulin growth factor 1 receptor on CTCs. We found that only 21% of patients had exclusively EGF-1 receptor negative CTCs. These patients had a very adverse prognosis. On the contrary, patients with insulin growth factor 1 receptor on CTCs, when treated with chemotherapy plus metformin, have a very good prognosis. Metformin is a diabetic drug. In this 50 slide, I hope to have shown you how it can be important to phenotypically characterize circulating tumor cells, and how it's possible to do it using the fourth channel. I would like to thank you for participating in this webinar, and I hope you found it useful for your studies. Bye!
Thank you so much, Dr. Rossi, for that fantastic overview of your research. And next, I want to introduce Dr. Jennifer Davids from Menarini-Silicon Biosystems.
Hi everyone, I'm Dr. Jennifer Davids and I'm the Director of Field Scientific Support for MSB. I wanted to speak today about heterogeneity in HER2 status. Are you getting the bigger picture? Briefly, this information is intended for biotech, pharmaceutical, and/or academic researchers. The products and services discussed are for research use only and are not for use in diagnostic procedures. As I'm sure you're familiar with already, the CellSearch system is the only FDA-cleared technology for the enumeration of circulating tumor cells, and is considered the gold standard with over 650 peer-reviewed publications. The platform has a lot of flexibility, and I want to speak today about the fourth channel. We have a coffee chat set up already for your viewing pleasure on open channel optimization that was recorded by Dr. Bamboo Dong, and it will briefly walk you through our optimization flowchart seen here. HER2 is known by many names. The human epidermal growth factor receptor 2 is also known as HER2/neu, ERBB2, and CD340. It's normally expressed on cell surfaces, but overexpression can lead to accelerated cell growth. It is one of the major canonical markers in breast cancer, along with the estrogen receptor and the progesterone receptor. HER2 expression and mutational status is generally assessed on tumors by a tissue biopsy. And you can look at these both with immunohistochemistry for protein expression and fluorescence in situ hybridization, or FISH, for locus amplification. We know that HER2 cancers tend to be more aggressive in phenotype. And HER2 status in breast cancer directly informs the decisions with HER2 targeted therapy or not. As you can see on the right, we can look at graphs of IHC for HER2 protein, and you can see the FISH staining that has HER2 copies as well. We know that HER2 is differentially expressed in a wide variety of tumors. So you can see here in yellow, HER2 amplification, in blue, HER2 overexpression, and in red, HER2 mutational status throughout a variety of cancers. In breast cancer, we generally find that HER2 is amplified in 15 to 20% of breast cancer tumors. However, we also know that CTCs, or circulating tumor cells, that are shed by primary breast cancer sites do not always have the HER2 amplification or expression status of the primary tumor. We have found that this non-concordance happens in about 5 to 19% of patients with breast cancer. So if you're treating patients who are HER2 negative but have HER2 positive CTCs, you may not be treating the entirety of the tumor. HER2 positive CTCs have been investigated as a biomarker to predict response to anti-HER2 therapies in patients with both HER2 positive and HER2 negative tumors. So we wanted to find out, with CellSearch, can we interrogate HER2 status alongside CTC enumeration in metastatic breast cancer? And the answer is yes. We have a CellSearch tumor phenotyping reagent for HER2 or neu, and you can see here in the right-hand column positive staining for HER2 in our normal CTC kit in the fourth channel. There's a lot of ongoing research in HER2 positive CTCs in breast cancer. This group worked out a formula to standardize HER2 expression, as you can see here. They also compared HER2 intensity in different cell lines, as you can see in figure A, and then compared that intensity with ICC and FISH results, as you can see in figures B and C. Using this scoring system, they were able to determine if a cell line or a cell from a blood sample was considered HER2 negative, as you can see in the top two cells here, or if the intensity was strong enough that they considered those CTCs to be HER2 positive. Since then, there have been some clinical trials that have looked at HER2 negative metastatic breast cancer patients who have HER2 CTCs. And the goal of these trials was to understand, for HER2 negative patients, if the presence of HER2 CTCs affected treatment. In the CIRCE-TDM1 trial, 155 patients with HER2 negative metastatic primary tumors were screened for CTCs. 104 of those patients were positive for CTCs, and of those, 14 patients were found who have HER2 positive CTCs despite having a HER2 negative phenotype at their primary site. This allows physicians to make more educated treatment decisions. In the DETECT trial, 105 patients with HER2 negative primary breast cancer but positive CTCs were enrolled and randomized into two different treatment groups. Half got standard therapy, and the other half got standard therapy plus lapatinib. At the time of follow-up, those patients who had no evidence of CTCs had a better overall survival compared to patients who still had CTCs present. The primary conclusion of the DETECT program is that HER2 overexpression on CTCs in patients with HER2 negative primary lesions provides a key signal to physicians to consider a more appropriate therapeutic option for their patients. Thank you for joining us today. If you have any questions, please feel free to email myself or our medical affairs team. Have a great afternoon.
Thank you so much, Dr. Davids. And thank you all still so much for being with us here today. If you missed any of the earlier talks, or if you'd like to share with a colleague, we will make them available after today. We have 2 more speakers for you today. I'm very pleased to introduce Dr. Paulo D'Amico from Northwestern University.
Good morning, ladies and gentlemen. This is Paulo D'Amico. I'm a medical doctor, and today I'm going to present preliminary results of our collaborative project that is going on between the Northwestern University and Università degli Studi di Milano. The title is Implementing a Novel Biomarker to Define the Role of HER2 CTCs in Metastatic Breast Cancer, the Circulating F2 Ratio. This project has been supported by the American Italian Cancer Foundation. So, since early 2000, we are using the CellSearch, that is the only FDA-approved machine, to enumerate and characterize circulating tumor cells in breast cancer patients as well as other cancer. And this is possible thanks to Professor Christopher Neely and other investigators that have demonstrated their prognostic value in 2004. We have been using 3 channels to enumerate CTC, but actually this machine gave us the possibility to further characterize circulating tumor cells with an additional antibody detection that is detected by the so-called 4th channel. The most relevant biomarkers has been tested using this channel in order to investigate the potential prognostic or predictive value of this further characterization of CTCs. But in this presentation, I will focus only on HER2, which expression in tissue is a well-established predictive factor of response to anti-HER2 treatment that, as we know, has positively overturned the prognosis of a subpopulation of metastatic breast cancer patients. So my question today is there any chance to identify through a liquid biopsy biomarker an additional population of patients that may benefit from this targeted treatment? But first of all, let's quickly review where we stand. So which are the clinical evidence supporting the use of HER2 expression or amplification in circulating tumor cells to recognize this portion of HER2 negative breast cancer patients based on tissue biopsy that may benefit from targeted treatment. This is the design of the CIRCE TDM1 trial that where HER2 negative metastatic breast cancer patients with HER2 amplified CTCs were treated with TDM1, that is an antibody drug conjugated against HER2. The overall response rate, that was the primary endpoint, was quite disappointing, only 11%. And there were other 4 patients achieving a stable disease by this drug in this contest among 11 patients. The supply actually of the FISH CTC assay discontinued the development in 2016, and the investigators had to stop the enrollment prematurely. So the curves that you see here are progression-free survival and overall survival, and as being compared with those that did not have HER2 positive CTCs and that were treated per standard of care. So you can deduce that actually they were definitely 2 different subpopulations treated with different therapies, so they were incomparable. This trial, in my opinion, has underlined the importance to have a well-recognized, a well-established, standardized method for CTC characterization. So we have the bad and we also have the good. In this case, this is a phase 3 randomized trial that has assessed the efficacy of lapatinib when administered in HER2-negative metastatic breast cancer patients that were bearing HER2-positive CDC. The preliminary results were published recently at the last San Antonio Breast Cancer Symposium, and we see that patients were randomized either to receive standard of care or standard of care plus lapatinib. This was the first phase 3 trial that have demonstrated a gain in terms of overall survival for these patients. We have a difference. If we administered lapatinib plus standard of therapy in this cohort of patients, we have a gain in overall survival. In this case, HER2 expression was assessed using the CellSearch, and only HER2 positive 3+ CTCs were considered HER2 positive. So there were a very strictly selection in patients. However, something remained unclear, because if we consider progression-free survival, that we know is a surrogate of overall survival, the curves are almost overlapping. So there is no difference. There is still something, in my opinion, in the process of understanding the role of HER2 positive CTCs that is missing. We still miss something. But what happens if we start taking in consideration the different intensities of HER2 expression in circulating tumor cells? What happens if we consider the heterogeneity of HER2 expression within CTCs of the same patients in the same blood draw? The same patient may have different CTCs with different HER2 expression with different intensity for HER2. So to address this question, and we know that almost the majority of the patients have this condition. There is very few patients has homogeneity of HER2 expression in CTCs when they have it. So to address this question, we created this new simple algorithm that we called the Circulating F2 Ratio, that is defined as the sum of 2+ CTCs and 3+ CTCs divided by the total number of HER2 positive CTCs. Using this algorithm, we both are taking in consideration CTCs with HER2 intensity that are different than 3+, but we are also taking consideration the mutual proportions, because we take in consideration how many 1+ a patient has and how many 3+, etc., etc. So please take note that the ratio goes up when we have few HER2 1+ CTC and goes down when we detect more 1+ HER2 positive CTCs. So we started from this point. Given the fact that the concordance between a liquid biopsy-based biomarker and the expression of the same biomarker in the corresponding tissue biopsy has been often used as a parameter of reliability of the liquid biomarker, we wanted to compare the circulating F2 ratio with the mere detection of F2 positive CTCs of each different intensity of expression with their power to predict an HER2 amplification in the corresponding tissue biopsy. So, are HER2 positive 1+, 2+, 3+ good CTCs to predict the HER2 positivity in tissue biopsy? The concordance is very low. We know that even if we consider only 3+ CTCs, the concordance with the tissue biopsy is low. But actually, we noticed that the circulating F2 ratio has a quite satisfying performance, definitely higher than the other biomarker assessed, identifying an HER2 positive disease on tissue biopsy, and we got an area under the curve ROC of 0.73. That is the best we got when compared with the other biomarker that we wanted to test the performance in this preliminary study. So notably, the performance is higher even if compared with the HER2 3+ CDCs. So, encouraged by these findings, we performed a retrospective study of metastatic breast cancer patients, 300 metastatic breast cancer patients that received a blood draw for CTCs analysis with CellSearch, and their CTC were stained for HER2. We selected those that were considered aggressive, so that had more than 5 CTCs in a tube, and we consider only the one that had at least one HER2 CTC of any intensity, so 1+, 2+, and 3+. Then we split the patients in two different subgroups of patients: the circulating HER2 high for those who had a ratio higher than 0.75, and the circulating HER2 low for the others, and we compared them. If we compare the distribution of subtypes among these two subpopulations, we notice that a higher percentage of circulating HER2 high patients is actually HER2 amplified in tissue, and we already knew that. 38% versus 19%. There is a higher prevalence of HER2 positive patients among these. But in order to confirm these findings, we collected data from circulating tumor DNA analysis and of these patients, and we compared the prevalence of HER2 alteration that we found on the circulating tumor DNA, and we found that surprisingly, half of the patients in the circulating HER2 high cohort had detectable HER2 alteration, compared with only 23% in the other cohort. So I will raise a provocative question now. Based on this evidence, which group would you think may respond better to an anti-HER2 treatment? Give your best guess. But what about the other population? What about the circulating HER2 low subgroup? They are characterized by a higher prevalence of endocrine receptor positive patients and a similar percentage of triple negative breast cancer. So based on this statement, again another question: which population would you expect to have a better progression-free survival to the next therapy? Probably the one with more endocrine receptor positive tumor that we know have a better prognosis, so the circulating HER2 low subgroup. Remember, these patients are the one that have more HER2 1+ CTCs. But if we compare the progression-free survival of the two subpopulations, we actually notice that the circulating HER2 low patients have a significant worse prognosis. So it's in contrast to what we thought before. So brief recap: patients with circulating HER2 high ratio have a better prognosis, are characterized by factors of favorable response to targeted treatment, and so on. Patients with circulating HER2 low ratio, they have more HER2 1+ CTCs, are more likely to be endocrine receptor positive, and they will probably be refractory to the therapy that we receive. But let's go deeper in the characterization of these two subgroups. Can this biomarker tell us more about the disease? Can we understand more? Can we know more using this biomarker? If we take a look at, for example, at the metastatic burden, that is both visceral in this case and not visceral metastasis, we find that the patients that are in the circulating HER2 low group, they are characterized by fewer sites of metastasis. So there is a fewer percentage of oligometastatic patients. In this case, we define oligometastatic patients as those who has less than 2 different sites of metastatic involvement. But what kind of metastatic behavior this population has? Can we identify a trend using this biomarker? Here we go. This is the visual involvement by site for both the populations. The circulating F2 low patients are affected by a disease that looks like seems to have a particular tropism towards specific site of metastasis. There is a higher incidence of liver, lung, and lymph nodes metastasis. Again, circulating HER2 low patients, more HER2 1+ CTCs, disease with multiple sites of metastatic involvement, tropism toward liver, lung, and vital organs. Worse prognosis. Is that enough to start thinking that we should give more relevance, more importance to this subgroup of cells of HER2 positive CTCs that have been so far neglected? Are they just HER2 negative CTCs, or they are actually a biological entity whose role should be addressed in future investigations? I would love to share with you where we are with our preclinical investigation toward this direction, but I suppose I ran out of time. So I want to thank you for your attention and stay tuned to this channel for the next episodes.
All right. Thank you so much for the wonderful talk. If you have any questions for Dr. D'Amico or any of our previous speakers, please type them into the chat window and we'll make sure to pass them along. Last but not least, we are very pleased to introduce Dr. Daniel Hayes, the Stuart B. Padnos Professor of Breast Cancer Research at the University of Michigan Rogel Cancer Center and former president of the American Society of Clinical Oncology. Dr. Hayes.
I've been asked today to talk about work we've been doing in my laboratory regarding characterization of circulating tumor cells using the CellSearch assay, going beyond enumeration or just counting cells. I'm fond of and obligated to transparently declare my conflicts. I certainly have conflicts regarding this talk because Menarini Silicon Biosystems has been a big supporter of my laboratory in regards of our use of CellSearch, both laboratory and clinical research funding. Furthermore, the University of Michigan owns a patent, and I'm the named investigator regarding the use of circulating tumor cells, especially phenotyping, and that is licensed MSB, and we receive annual royalties. I have a number of other conflicts which are not related to this talk. Let's start out with some definitions. In the word liquid biopsy is, the term is commonly used now principally to refer to ctDNA, but in fact, that's not correct. The term liquid biopsy was in fact coined in 2010 by Klaus Pantel and Kathy Alix-Benabriere, who, as defined as, research groups are focusing on the clinical value of CTC analysis, and although promising data from patients with advanced disease demonstrate the value of CTC analysis as a liquid biopsy, studies on cancer patients at earlier stages are hampered by low CTC counts. That's all true, and in many respects, Drs. Pantel and Alix-Benabriere were foreseeing the advent of using circulating tumor DNA, but in fact, they were really referring to CTCs when they said liquid biopsy, and I think that's an important issue, that liquid biopsies really refer to anything we could find in blood that might tell us the status of the cancer. Now, I believe that both liquid biopsies and tissue biopsies will be complementary. In terms of liquid biopsies, they're obviously easy to draw. Phlebotomy is relatively inexpensive, and it permits really easy serial testing, whereas tissue, of course, is invasive. You have to really do a biopsy. It's more difficult to obtain. You have to use interventional radiology. It's often very expensive. A pathologist usually needs to be involved, and serial testing, therefore, is logistically quite difficult. The pre-analytical issues of circulating markers are easier to control, fixation, anticoagulation, et cetera, all in a single vacutainer tube, whereas the processing of tissue can be quite complex and relatively easy to screw up, frankly. On the other hand, the sensitivity of liquid biopsies is much lower than what you see in a tissue biopsy. CTCs, and for that matter, plasma tumor or circulating tumor DNA are both really in low abundance, with CTCs being extraordinarily rare events. Whereas, of course, when you do a biopsy, you get millions or even billions of cells in a single needle biopsy. Finally, though, there may be some complementary issues regarding the biology. A liquid biopsy really tells you what's going on in the entire organism, not just the site where you do the biopsy. On the other hand, what's in the blood may not represent the biology of the tissue-based cancer, and vice versa in regards to tissue. So I don't believe any of these is going to replace the other, but rather that they'll be complementary in our regards to taking care of better patients. I'm briefly go over CellSearch. There are really now well over 40 different capture platforms in the English literature, probably more than that. CellSearch is the one that is really at the forefront. It was the first FDA-approved assay for circulating tumor cells and remains really one of the most widely used assays. The pros of CellSearch are it's highly analytically validated, it's highly automated, it's modestly flexible, and the clinical validity, which I'll show in a minute, has been strongly demonstrated. The cons are really less sensitive than desired. It depends on EpCAM capture. You lose CTCs in the process. And again, we're limited in the amount of blood we can draw in a single patient. And so there are probably ways to revise this to make it even better. But right now, CellSearch is one of the better games in town. And this is the closest thing I have to an advertisement. These are just the pieces of equipment and supplies we use to run our assays. Now, most of you probably know how the assay is done. It's based on immunomagnetic particles that are in suspension, and those are bound to anti-EpCAM antibodies. So anything that's making EpCAM gets pulled down by a magnet, and then you come back in with anti-cytokeratin, anti-CD45, and DAPI. DAPI to prove it's a nucleated event, cytokeratin to prove that that event is indeed epithelial, and CD45 to prove that it's not leukocytes. To be clear, CellSearch does not purify CTCs. It enriches them from the billions of red cells and millions of white cells that are in 7 to 10 mils of blood. So then you put it through a laser, and the laser captures each of these things, and it shows you this is a nucleated event. It's an epithelial nucleated event because it's cytokeratin positive. It's negative for CD45, and it puts these together in a composite that a technician needs to go through and say, yes, those are cancer cells, or no, they're not. But importantly, there's a fourth channel. It was originally developed for calibration, but the machine is really highly reproducible, and we found doesn't need calibration. So we can use that fourth channel to do additional phenotyping as long as we can fluorescinate an antibody, that the antibody maintains its activity with the original protein, and that that protein is membrane bound either within or on the surface of the cell. These are data just regarding enumeration. These are old studies dating back now 17 years when we published our first paper in the New England Journal regarding CTCs in breast cancer, but you can see they are highly prognostic in breast cancer, colorectal cancer, prostate cancer, and then more recently lung cancer. And these have been validated over and over again as a highly prognostic marker. Furthermore, in a study we did in SWOG, what we found is not only is it prognostic at baseline, it's even more prognostic in telling you whether that patient is likely to be benefiting from the therapy he or she just started. These are patients who started first-line chemotherapy in SWOG SO500, run by a colleague of mine at Michigan Medicine, Jeff Smiraj. And the blue line are people who did not have elevated CTCs at first follow-up. These are all metastatic breast cancer patients. The yellow line are patients who had 5 or more CTCs and cleared them within 1 cycle of chemotherapy, meaning they'd had a CTC response. And the gray line are patients who had elevated CTCs and did not clear them below 5 CTCs per 7.5 mils of blood, meaning they did not have a robust CTC response. And you can see that overall survival is clearly different in these 3 groups. Sorry, I meant to do that. So that for arm C, the group of patients who did not appear to have a CTC response, half of those patients were dead within 13 months. 3/4 of those patients died of their cancer within 18 months. And again, this is first-line chemotherapy. So this is a very poor prognostic category. However, in this era of precision medicine, we'd really like to do more than just count cells, but actually look at their biology and see if we can use the markers that we commonly use or new markers to identify targeted therapies that might help us do even better. Let's talk about phenotyping first, and that's where we use the fourth channel. What we found is that we can use that fourth channel for a number of markers. And again, the criteria are just that the antibody has to survive fluorescence and bind to its antigen, and that antigen has to be membrane bound. It can be internal membrane, nuclear membrane, for example, or it has to be cell surface membrane, as long as it survives fixation. Now, this is a shopping list of a number of proteins that have been reported. The yellow bar are ones that we've done, I'm going to talk more about, and the papers to the right that we've published, principally led by a very, very talented woman in my lab, a medical oncologist who worked in my lab for almost 10 years, Costanza Pelletti, who really pushed this field out to where it is now, in my opinion. The blue is for BCL2 and apoptosis, and again, we've published a couple papers regarding these. And the final one, the green, is PDL1, which we're working on now. We have a paper submitted, but it's not yet accepted or in press. We personally have done IGFR1, androgen receptor, and vimentin, but we haven't published anything on these. Others have. So as an example, the first question we asked was, could we use these phenotyping in the fourth channel to identify women with estrogen receptor positive metastatic breast cancer who will not benefit from the next endocrine therapy? I think most of you know the paradigm for ER positive metastatic breast cancer is to treat patients with serial endocrine therapies, each until it quits working, and then go to the next one. And finally, when a patient appears to be refractory to all endocrine therapy, you move on to chemotherapy. The question is, can we find patients early who are not going to benefit from any endocrine therapy and take them on to chemotherapy where, although it has more side effects, it is probably more likely to be beneficial? So we reckoned that we could use these 4 genes to identify patients who either had sensitive or resistant disease. Estrogen receptor, of course, and also BCL2 probably predict sensitivity to endocrine therapy, whereas HER2 and Ki67 probably predict resistance. And you can see the references below that I've got here that back this up. We then worked very hard to develop an index of reading for each of these markers. And so what we did is we ran a pilot trial of about 50 women. We had to draw a different tube of blood for each of the markers, one for ER, one for BCL2, one for HER2, and one for Ki67. So that's roughly 8 mils of blood times 4, 32 mils. We would pool those and then split them back up into 4 7.5 mil aliquots. You can see here the number of cells per aliquot is pretty close. It's within the variation of the assay. And down below, you can see in this particular patient who originally had ER positive and HER2 negative breast cancer, her CTCs were positive, her BCL2 was positive, her HER2 was negative, and her Ki67 was negative. On the left-hand side, the composite are the actual CTCs, and then that second column is the marker. So that very black background and very white staining implies positivity. And you can see you've got positive ER, BCL2, and positive Ki67 in this case, but lots of gray background and just a smudgy cell really indicates that that was a negative cell for HER2. And from this, we developed a so-called CTC endocrine therapy index, or CTC ETI, with the feeling that the more cells you had, and especially the less likely they were to be ER positive, and the less likely they were to be BCL2 positive, but the more likely they were to be HER2 positive and have elevated Ki67, the more likely they would be to be resistant to endocrine therapy. This particular patient had ER positive HER2 negative breast cancer. She had more cells per tube than the last patient you can see on the top. And what you see here is that in this case, although she had plenty of cells, each of which looks like the first column there, none of these cells was positive for estrogen receptor. So we assigned that patient a very high CTC ETI on the basis that she was ER negative. Her cells were BCL2 positive, HER2 negative, but also elevated for Ki67. So we would predict that this patient might not do well on endocrine therapy. We, as I noted, did almost 50 patients. And Dr. Pelletti worked out this scale on the right so that cells that had no staining in yellow were considered negative. Those that had little staining in pink were considered 1+. Those that had some staining in blue were 2+, and those that were very bright with very dark background were 3+. And what you see here on the top is a series of patients, 11 to 51, it doesn't matter, it's just that patient number, and the number of cells they had per 7.5 mils of blood. And down the bottom, for each of those patients, you can then see the stack of different ER in those cells. What you also see is, except for a few examples where patients were completely ER negative, their CTCs were all ER negative, on the most part, there was great heterogeneity within each patient. I want to point out this patient in particular had almost 1,000 CTCs per 7.5 mils of blood. That's very high. She did not do well. But I also want to point out the enormous heterogeneity that she had, so that in her CTCs. So about 1/3 of her cells were ER negative. About another 11% were only 1+. We consider completely negative or 1+ to be negative. And then about half of the cells were 2+, and then 3% were 3+. On the right-hand side, you see an immunohistochemistry of this patient's original primary, where 95% of these patients were very bright ER. So clearly, at least by comparing CTCs when she developed metastasis compared to her original primary cancer, she had developed ER negative clones that were really not very visible in her original primary cancer, consistent with the fact that she was indeed refractory to endocrine therapy. So based on these preliminary data, we started a prospective study of about 120 patients who were starting second or third line endocrine therapy for metastatic breast cancer, using this so-called CTC endocrine therapy index. Again, Dr. Pelletti ran this trial, and the results of this trial are now in press. We expect to be published within the next month. Unfortunately, our hypothesis didn't really pan out as well as we thought. On the left, you see outcomes related to baseline CTCs, with the black line being less than 5 CTCs per 7.5 mils of blood, and the red line being greater than or equal to 5 CTCs elevated. And what you see is we expected about 1/3 of these patients had elevated CTCs. That's what we've seen over and over again in ER positive patients. But you can also see that the survival for patients with not elevated CTCs was not quite, but almost twice as great as those who had elevated CTCs, with a statistically significant p-value. What you see on the right-hand side is the same set of patients, but this time broken into CTC ETI. The black curve is identical on the right as it was on the left, because all of the patients who had less than 5 CTCs were considered to have a low CTC endocrine therapy index. But the red line on the left has been broken into those patients who had elevated CTCs, but also had a poor biologic finding in their CTCs, versus those who had elevated CTCs, but didn't have such a bad, basically had a CTC ETI low. And while visually there is some difference between the red line and the green line, statistically there is not. And part of it is because so few patients fell into the low intermediate group. Most of the patients who had elevated CTCs also had a CTC ETI that was high. And you can see that the prognosis in the upper right-hand corner there is worse numerically, but it's not worse statistically for the green and the red. We're still interested in that. We're still pursuing it further to see if there are ways we can tweak the assay and make it better. But we've also recently developed genotyping. We and others have begun to be able to not only enrich the CTCs using CellSearch, but then get them to purity by separating them completely from any contaminating white cells using an assay called dep array, and then taking the cells, amplifying the DNA from each cell, and sequencing them. We hypothesized that a liquid biopsy using circulating tumor cells or circulating plasma tumor DNA would facilitate assessment of somatic mutations above and beyond what you might do with a biopsy, and we have also hypothesized that they could be complementary. And to show you one example here, this is a patient. The columns are individual CTCs. The rows are individual genes. The first two are ecadherin, and the last three are estrogen receptor, the genes that encode for estrogen receptor and specific mutations. Dark green means a homozygous mutation, both alleles, or one allele deleted and the other mutated. The light green means that one allele was wild type and the other allele was not, was mutated. NC means that we were unable to determine for that particular gene, for those particular CTCs across the board here. And on the far right, you see the tissues. So this patient was felt to have the 2 mutations in CDH1 and to have a Y537S mutation in ESR1. But we also found a mutation that had not been reported before at A569S. In a single cell, but we're pretty sure this was real and not a contaminant, and that was not seen in the patient's tissue. So I've been talking through those. We also found one cell in this data set that was completely wild type, had no mutations at all. We think probably this is a normal epithelial cell that was isolated. Why would there be a normal epithelial cell in the blood? We don't know. We noticed that a long time ago when we first started doing CellSearch. There are occasional wild type circulating epithelial cells that get in the blood for who knows what reason. But it also needs to be kept in mind. And finally, this is a heat map of copy number variations, not mutations. So again, each column in this case is a different gene, and each row is split into patients. So the black line separates each patient, and then each row within the patients is a different CTC. So patient 2, patient 4, so on and so forth. And what you can see just by looking at this is enormous heterogeneity, both between patients, but also for individual genes among the different CTCs. And I want to highlight this particular one. This is ESR1, so of interest to us. And if you just look at patient 2, the top patient, what you can see here is that a couple of her cells had deletions of ESR1, a couple of her cells had amplification of ESR1, and others were wild type for copy number variation within the same patient. This is why we don't cure metastatic breast cancer, because of this sort of heterogeneity. And these are the kinds of things that we're moving forward in now in prospective studies to see if we can begin to identify this kind of heterogeneity and these clones and attack them in a smart way instead of empirically. So I'll end up with this slide. I personally believe that tissue, CTCs, and ctDNA will be complementary for all these reasons, is that you get a lot more cells with tissue, so you could do a lot more genotyping. And you can do candidate assays with CTC. It's a little harder to do with ptDNA. We know that you can phenotype tissue and CTCs. You cannot phenotype ptDNA. Tissue does not represent the whole body. It only represents the area where you stick the needle, whereas liquid biopsies do represent the whole body. We know that tissue represents tissue biology. We've known that for 50 years. The question is, do the circulating markers represent tissue biology and give us insight into patient outcomes and clinical care? And we also know that it's difficult to do serial tissue biopsies. It's easy to do serial liquid biopsies, and that's where we're going with our research. So to conclude, enumeration is highly prognostic at baseline. It's even more prognostic after 3 to 4 weeks of treated. In my opinion, it's a so-called retrospective predictive factor, because it gives you an early insight into how that patient is doing on that therapy. Phenotyping is possible. There's potential is enormous here, but presently it's still investigational. And genotyping is also possible. Again, also presently investigational, but will give us great insight into tumor heterogeneity. So thank you very much. I'm sorry I can't join you in the day that this will be shown. Be happy to answer any questions by email or any other way we can chat. These are the folks who really did the work, especially Dr. Paletti, who has now gone into industry last summer. Emily and Elizabeth, Emily Doltz and Elizabeth Darga are technicians in my lab who did most of the hard work. And then on the right, Scott Tomlin's a great collaborator in our Department of Pathology, and Andy Canny, who did a lot of the genetics and is now a postdoc in my lab. These are the people who supported the work of the BCRF, the Susan Komen, and then AstraZeneca and Menarini. Thank you very much.
All right. Thank you so much to Dr. Hayes. That was our last presenter of the day. Thank you all so much once again for attending, and thank you very much to our amazing speakers. Thank you, everyone, for your patience as we work through some technical issues. We hope you had a good time, and please let us know if you have any questions for the speakers or if you have any questions about the CellSearch system or our lab services. Thank you again, and may the 4th be with you.